I will share my screen.
Okay. All right. So welcome, everyone. Thank you for joining us today at our December technical seminar. So this is a monthly seminar series jointly hosted by myself from the Experimental Medicine Graduate Student Society, as well as the societies from Physiology and Pharmacology and Therapeutics. And so the idea is for students to teach other students about the techniques that they use in the lab, from data analysis to wet lab techniques and everything in between. And so today I'm really excited to introduce Neelima, who is completing her PhD in the Cloutier Lab at the Montreal Neurological Institute. And today she'll be discussing the applications of CRISPR-Cas9 to study neural circuit formation and speaking in depth on how to generate and validate a CRISPR mouse model. And so with that, Neelima, I'll let you take it from here.
Okay. Thank you for this opportunity. And today I'll be talking about generation and validation of CRISPR-Cas knock-in mouse to understand the role of KRL3 receptor in axonal targeting. So before I begin, I'd like to emphasize that some of the results I'll be sharing today stem from a close collaboration between our lab and the Oskin lab at University of Chicago. So we rely on various traffic signs in our day-to-day life to get to our target destination. Similarly, neurons in the brain rely on various guidance cues or proteins that serve as traffic signs to make accurate connections in the brain. But the question is, what are these proteins and how do they help neurons make accurate connections? Now, if you think of a scenario where you're in your house and the lights are out and you smell freshly baked cookies in the kitchen and you want to get there. So first you smell the cookies, so you're walking the direction of the smell, and you're also physically touching objects in your immediate environment to navigate. So in this example, the smell of the cookies was a long-range cue, and physical contact with your immediate environment is a short-range cue. So neurons in the brain use a very similar strategy. They rely on various long-range cues and short-range cues that can be attractive or repulsive in nature. And one such example of a contact-dependent short-range attractive cue is the KRL family of proteins. Now, this family constitutes 3 members, KRLs 1, 2, and 3. And in my project, I focus on KRL 2 and 3. They've been shown to possess a homophilic cell adhesion property, meaning KRL2 binds to KRL2 and KRL3 binds to KRL3 in vitro. It's been shown in vitro. And they've been shown to play a role in synapse development and circuit formation in flies, worms, and mice. Within the mouse model, they've been shown to play a role in synapse formation in the hippocampus and the olfactory system. And I focus on the olfactory system. So if we take a close look at this olfactory system, it can be subdivided into the main olfactory system and the accessory olfactory system, both of which are crucial for the survival of mice and modulating various social and sexual behaviors in mice. So if we zoom into these two systems, we see that they're very similar in their broad organization. I will be focusing in this talk on the accessory olfactory system. What you see here is the system has sensory neurons called vomeronasal sensory neurons, whose cell bodies reside in the nasal epithelium called the vomeronasal organ, and they project their axons to the brain, in a region in the brain called the accessory olfactory bulb, or the AOB, and they synapse onto the dendrites of second-order projection neurons called mitral cells in the circular structures called glomeruli. Now, each sensory neuron expresses a certain type of receptor. All neurons expressing the same type of receptor grouped together into common targets called glomeruli. Now, these concepts are nicely exemplified in the circuit diagram here, where you have sensory neurons projecting from the nose to the brain, and you have various long-range cues allowing the segregation of these axons into posterior AOB or anterior accessory olfactory bulb. And you have some short-range cues. Once they segregate into their broad targets, they allow the fine-tuning of these axons. And as I mentioned, KRLs are a good example of short-range cues. Now, KRL 2 and 3 are expressed along the these axons, different populations of axons expressing different receptors in different amounts and combinations, forming a barcode or a profile. So you have, depending on the combination of KL2 and 3, you have 4 possibilities of different combinations, and all sensory neurons grouping together into a common target glomerulus have a similar KL2, KL3 expression profile. And you can see that in this example here. This is a section of the brain. In green, you see KL3, and red, you see KL2, while and in these circles, you see the ends of the axons coming together. So some axons have KL3 and KL2, where express KL3 and KL2, whereas some axons express only one or the other. So if these molecules serve as a barcode for the grouping of like axons, when you ablate one or the other, you should expect defects in axon targeting. And that's exactly what we see here. When you knock out KL3, you see fewer and larger glomeruli, indicating that KL3 is important in this process. Similarly, we see that for KL2. So far, I mentioned that KL2 and KL3 are important for axon sorting in the accessory olfactory system. Previous studies from our lab and other labs have shown that KLs play an important role in axon targeting in the main olfactory system. I mentioned to you that these KLs have a homophilic cell adhesion property shown in vitro. But the question still remains, is the homophilic cell adhesion property required in the vomeronasal sensory neuron axon sorting and coalescence in the accessory olfactory bulb? To answer this question, in collaboration with the Oskin lab, we looked at the crystal structure of KL2 dimers and KL3 dimers. And in this dimer, at the binding interface, we saw we were able to identify various amino acids that might play an important role in the dimerization. So with this information, we introduced single residue mutations in KL3 that could potentially disrupt dimerization. And using an assay that separates molecules based on size, we tried to identify whether dimerization was disrupted. And what was found is that this Q128A mutation, glutamine to alanine, had completely disrupted the dimerization of KL3. And this was further verified using a cell aggregation assay. So cells in culture were transfected with wild type KL3 that's GFP tagged, or the mutant KL3 that's GFP tagged, and was observed. And what they observed was in the wild type KL3, there was aggregation of cells, indicating that the homophilic cell adhesion property was intact. And in the mutant KL3 cells, you did not see aggregation, showing that this clustering of cells was disrupted. So with this information in vitro, I disrupted cell adhesion and generated a knock-in mouse with this Q128A mutation in vivo. And for that, I used a technology called CRISPR-Cas genome editing technology. Now, brief overview of, I'd like to give you a brief overview of what CRISPR-Cas is. As some of you would, and most of you would know, that it was first discovered in bacteria as an immune mechanism, immune system mechanism. So what happens is you have, when viruses invade bacterial cells, a part of their genome is chopped up and incorporated into the bacterial genome, and that's used to transcribe something called the tracer RNA. And you have something else called the Cas9 enzyme. And when the virus, the same virus invades the cell again, there's a complex formed with the viral genome, the Cas9 enzyme, and the tracer RNA, and the genetic material of the virus is chopped up and degraded. So this is used to our advantage as a gene editing tool in animal models for generating whatever manipulations we want. So in this system, how this works is, let's say this is your DNA of interest, and this is your target site. This is where you want to generate, introduce a mutation. And you have your Cas9 enzyme, your single guide RNA, and your donor DNA. So this forms, and so basically your Cas9 enzyme and single guide RNA form a complex with your DNA of interest. Now your single guide RNA recognizes something called a PAM site, which is basically NGG, nucleotide GG, and the N can be anything. So here it's, let's say it's AGG, and there's different AGGs here in the sequence, as you can see, but each single guide RNA recognizes a specific PAM site. So the PAM site and the sequence surrounding it. So let's say your complex of Cas9 and single guide RNA is formed here, and the cut is made over here. And what happens is 100 base pairs on either side of the cut are chewed up by repair mechanisms, and this entire stretch is going to be removed. And once the stretch is removed, you have homologous, sorry, you have HDR, which is homology-driven repair, or NHEJ, which is non-homologous end joining repair. So when you have a donor DNA filling in that gap, you introduce, if you have a donor DNA filling in that gap, it's going to be, sorry, can you hear me? Okay. The gap is going to be filled up with random insertions and deletions. So as I mentioned, we are using this homology-driven repair to introduce a knock-in mutation. So a quick recap, we need a Cas9 enzyme, we need to design efficient single guide RNAs, we need a donor DNA, and what's to introduce a restriction enzyme site with a silent mutation so that it doesn't affect the coding of the protein, to further verify whether the donor DNA was incorporated or not, for screening purposes. So to design efficient single guide RNAs, what do we need? One, we need to ensure that there are minimal off-target effects. We need to factor in how close the single guide RNA is to the target site, because they're saying that the guide is a silent mutation in the PAM site of the donor DNA. Why? Because once your donor DNA is incorporated, you don't want the complex formation and cutting to happen over and over again. So once the donor DNA is incorporated, if you have a silent, and we need to know what the cutting efficiency of the guide is. So to design efficient guides, first we identify the FASTA protein sequence of interest, in this case KIRL3. We identify the FASTA KIRL3 DNA sequence. We do a protein to nucleotide blast and align the two, and identify 100 base pairs. So we identify the DNA sequence that corresponds to the amino acid of interest in the protein, and identify 100 base pairs on either side of those sequence, those nucleotides, and use that to input in a software. So in this case, 100 on either side, and the 3 nucleotides coding for that amino acid, 203 base pairs, and you input that in this MIT sgRNA tool, or you can also use something else called Cas9. So once you input your sequence, you will get a list of RNAs that are in the target site, other factors that I discussed, and of this. First, once you choose your single guide RNA, you need to generate it. Knowing the sequence, you design primers that overlap like this, and you do this. You run a PCR so that these ends are completed, and you get a DNA template for your single guide RNA, and you do in vitro transcription and generate your single guide RNAs. So once you've generated your single guide RNAs, it's important to test for the efficiency of these guides. And the way you do it is, I mentioned that there's something called non-homologous end-joining repair. So we use that principle to our advantage here. So we don't introduce a donor DNA. We use this system, and what we do is you micro-inject Cas9 enzyme and single guide RNA only in a mouse embryo that's at the 2 or 3 cell stage. They say single cell, but for all practical purposes, there's 2 or 3 cells at that stage. So you don't inject a donor DNA. So what happens is when the DNA, the region is chewed up, you have a gap which is filled up with random insertions and deletions. So what you do is you let these this embryo grow to the blastocyst stage in vitro, and then you extract the guide RNA. If at the target site, you would expect insertions and deletions using this method. If you don't see insertions and deletions, it's not cutting. Another method to screen, it's an indirect method, which is the same thing. It involves injecting with Cas9 and single guide RNA, collecting the blastocyst, extracting the DNA, and then there's something called the T7 assay. So what this involves is basically when you first inject the Cas9 and single guide RNA in the 2 or 3 cell stage, some cells take up the complex and some cells don't. So some cells will have random insertions and deletions and some cells won't. So when you allow it to grow to the blastocyst stage and you collect the DNA, you will have a mixture of DNA with insertions and deletions and without. So then you use a sample to heat up this DNA so that the duplexes unwind and form separate out, and then you cool the sample so that they come back together randomly. So that allows formation of heteroduplexes. And when you have heteroduplexes in this assay, there's an enzyme called T7 endonuclease that identifies heteroduplexes and cleaves them. And when you run on a gel, you see 2 bands because of the cutting of the DNA. So if there are no heteroduplexes, it would mean that the cutting there's no random insertions and deletions in some of the DNA. That indicates the efficiency of the single guides. So using this method, I shortlisted 3 guides from that long list that I got from the MIT guide RNA tool. So these are just numbers on that list: guide 4, guide 8, guide 28. So I shortlisted these guides, and some of them the reverse guide, 2 of them were forward guides. The step is to generate to design the donor DNA. So for the donor DNA design, what's important is that your donor DNA has a restriction enzyme site with a silent mutation for screening purposes. It has a silent mutation in the PAM site so that the cutting doesn't happen again. And after the donor DNA is incorporated, and a mutation, of course, at the target site, mutation of interest at the target site. So in this example, my guide RNA is a forward guide. So it binds to the reverse strand, and the cut is made such that the forward strand of the DNA complex is set free. And this means that the single-stranded donor DNA needs to be complementary to the foreign. And with all these requirements incorporated, you generate your single-stranded donor DNA, which will then be used to fill up that gap. So what happens next? First, we micro-inject Cas9, single-guide RNA and donor DNA into, this time the donor DNA, into the embryo at the 2 or 3 cell stage. We implant it into a female mouse, let the female mouse give birth, and the pups are then screened for the presence of the donor DNA. So what you do essentially is you use restriction enzymes to check for the presence, you check for the presence of the knock-in. So if you have a restriction enzyme digestion, you'll see a band that's a lower molecular weight than the undigested sample. And that would tell you that the donor DNA is incorporated. So we have the donor DNA, we have efficient single-guide RNAs. We've tested for the efficiency, and we've verified whether the knock-in has happened or not. So one important thing you need to note here is that when you inject this complex, as I mentioned earlier, into this 2 or 3 cell stage, and when you have a knock-in mouse, not all cells take up this complex. So not all cells will have the mutation. So this mouse that's positive for the knock-in is called a founder mouse. And what's important is that the mutation needs to be incorporated into the germline or the sex cells, so that when they are made to breed, the future offspring will have the mutation. So what you do is you cross the founder mouse with a wild type mouse. So as you can see here, you would expect if your germline in the founder mouse has the mutation, you would expect all the offsprings to be heterozygotes for that mutation. And my we had 2 heterozygotes, such that you would expect one wild type when we crossed 2 of the heterozygotes from one of our founder lines. So, okay, so great. We generated a CRISPR-Cas knock-in mouse. The next step was to validate this mouse. And the one method we used to validate, we used 4 different methods to validate our mouse. The first one was sequencing. So as you know, I can just quickly run through this. In sequencing, in single sequencing, essentially you have these DDNTPs that stop the reaction wherever they're incorporated in as the sequence is being made. And depending on where they're incorporated, you have different length fragments generated. So when you run these fragments on a gel, you'll see, and they're fluorescent. So you'll see different fluorescent bands. Each band corresponds to a certain nucleotide, and that's how you sequence. The other method is the restriction enzyme method. So what we expected, the way I designed my restriction enzyme site, is that the original length of the DNA was around 300 base pairs, and the restriction enzyme site was somewhere in between. So you expected 2 pieces that were around 150 base pairs, 180 and like 140, so close to 150 base pairs. So you expect in a wild type, you don't expect any restriction enzyme digestion because it's a wild type. There's no knock-in. So the band is the same as undigested. And in the knock-in mouse, the full knock-in mouse, you expect a band at around 150. And undigested, you expect a band around 300. And in the heterozygote, you expect two bands, one at the wild type level and two at the knock-in level, because there's two alleles, one wild type and one knock-in. So using this, we got these results. So with the sequencing, we ensured that the CAG to GCC mutation was indeed present. And using restriction enzyme, we confirmed that we expected the appropriate bands, or we saw what we expected for each genotype. So the next thing we did was verify the total protein levels between the wild type and the knock-in mouse. And we did this using Western blot analysis. So a quick refresher, we denature proteins, we extract proteins from the brain, lyse the tissue, denature the proteins, run it on a gel. We use charge to separate out to a nitrocellulose membrane, again using charge, and then use immunoblotting. So we have a primary antibody binding to the antigen, a secondary antibody tagged with an enzyme binding to the primary antibody, catalyzing a reaction. When you add a substrate, it catalyzes a reaction, generates a product, and that product gives you a signal so that you can visualize the bands. And when we used Western blotting, we saw that there was no difference between wild type and knock-in mouse in terms of total protein levels, as that's what we expected. And the next thing we did was we verified whether this CRISPR-3 Q128A protein is trafficked to the cell surface. So CRISPR-3 is a cell surface protein, and we wanted to ensure that introducing this mutation that disrupts homophilic cell adhesion property doesn't affect the trafficking to the cell surface. So for that, we used an assay called cell surface biotinylation assay. So in this, what you do is you take sections of the brain, and then you incubate those sections with biotin at a low temperature. So what happens is at a low temperature, biotin only binds to all proteins at the cell surface, not intracellular proteins. It doesn't go inside the cell. So then you lyse the tissue and release all the proteins, some that are biotinylated at the cell surface and the intracellular proteins. You incubate this lysate with streptavidin beads that bind to biotin. So when the beads pull down proteins, they only pull down biotinylated cell surface proteins. So there's three sets that we make. One, the pull down, which is the biotinylated lysate and the beads. So this we expect just cell surface proteins, no intracellular proteins. The next is we don't biotinylate some tissue, and that's the negative control. And when we run that lysate with beads, we shouldn't expect any proteins because we didn't biotinylate the tissue. The third is we keep some total lysate aside, not pull down with the beads, but the whole lysate that includes the cell surface biotinylated proteins and intracellular proteins. That's our positive control. So what I did was I perfused the mouse. I dissected the brain, sectioned the brain sagittally, and incubated with biotin, lysed the tissue, incubate the lysate with streptavidin beads, spin down the beads, and then suspend the beads in sample buffer and do a Western blot analysis. Took some of the lysate aside, reserved it as total lysate. So when I did this, I saw that this avidin pull down for wild type, there was an avidin pull down band for wild type and for the Q128A mutant. And in our positive control, the total lysate, we see and in turn, the total lysate, but not for the pull down as expected. And we see a stronger band in the total lysate. So this tells us that CRISPR-3 is trafficked to the cell surface, even in our Q128A knock-in mice. And the last validation was to make sure that KIRL3 is expressed along the sensory neuron axons using immunohistochemistry. So in this technique, you perfuse mice. So perfusion is essentially you flush PBS through the blood vessels and you drain out the blood. So you pull out the cap on the right atrium and flush PBS through the left ventricle, and the liver turns pale. So once you flushed out all the blood to prevent autofluorescence, you fix the brain by injecting PFA, paraformaldehyde, through the system, let it circulate, dissect out the brain, cryoprotect it to prevent damage from freezing, and then you freeze the brain and you collect sections from the brain and use immunofluorescence. So what happens is your primary antibody binds to your antigen, your secondary antibody tagged to a fluorophore binds to your primary antibody and fluoresces the color of interest. So using this, I looked at KIRL3. So in green, you see KIRL3 along the sensory neuron axons. These are basically the ends, the axons coming together in the accessory olfactory bulb. In blue, you see it's a counter stain. And yeah, so this confirms that KIRL3 is present along sensory neuron axons. So this is all the results from my validation. So sequencing, Western blot, cell surface biotinylation, and IFC. So now we have a validated knock-in mouse trait, but what are the defects observed? Are there any defects in the accessory olfactory system? What does it mean in terms of axon targeting? So for that, we went back to the accessory olfactory system, took sagittal sections of the brain and did an IHC for VGLU2, which is a marker for these axons. And what you see here, these individual clumps are basically clumps of axons in the brain, the ends of these axons in the brain. So I'm calling them glomeruli. So I manually counted these individual units of green in the brain. And what I observed is a significant reduction in the number of glomeruli and a significant increase in their size. So therefore, for the first time, we show that homophilic cell adhesion is required for KIRL3 function in axonal coalescence. So to summarize, I'm looking at the role of KIRLs in axon sorting in the accessory olfactory system. The system, depending on what receptor these sensory neurons express, there are different populations of sensory neurons in the system. So pink is one population, blue is one population here. And each population homogeneously expresses a certain combination of KIRL2 and KIRL3. So let's say the pink population is expressing high KIRL2, low KIRL3, and the blue population is expressing high KIRL2. So we know that KIRL2 and KIRL3 have a homophilic cell adhesion property, but we ask whether this property is important for the grouping of like axons that are similar. So we disrupt homophilic adhesion in vivo. And what we observe is that there's a heterogeneous mix of axons grouping together to form fewer and larger glomeruli. And the reason here being, what we propose is that the contribution of KIRL3 to this molecular. So instead of having high KIRL2, low KIRL3, the meaning of this is you only have high KIRL2 when you disrupt KIRL3 adhesion. So its contribution to this code is lost. And now both these populations that were otherwise different now seem similar, and they group together into a common target, forming fewer and larger glomeruli. So with that, I'd like to thank everyone involved in this project. It's not mentioned here, but a special shout out to my lab mate, Kat, who helped me with a lot of the initial CRISPR experiments. And I'm open to any questions.
All right, thank you so much for your talk, Neelima. It was a very clear and comprehensive overview on how we can use CRISPR-based technology to study biological functions. And I thought that the neural circuit context was also really interesting. Before we jump into questions, I just want to mention that Neelima has kindly created a step-by-step guide for generating CRISPR knock-in mouse model, and you can find the link for it on our website in the chat. And if you find the schematics useful and you want to use it, I just ask that you contact Neelima before you do so, as it is her work. So with that, are there any questions for her? You can raise your hand or type it in the chat. I mean, I can start off with one. So you were talking about the cell surface biotinylation assay to look at the cell surface expression of, I guess in your case, CRISPR-Cas9. I was wondering what the benefit of that assay is over something like flow cytometry.
I'm not too familiar with flow cytometry, but can you emphasize on what that technique is?
So like you can label cell surface proteins with antibodies, and then you essentially put it through the flow cytometer, and then it can detect like single cells that have cell surface expression of that antibody, basically.
Okay, I guess you can use that as well then, but in our case, this is also like in literature, it's the most commonly used method to show cell surface expression. It's in slices. So in like tissues that of interest, you can collect tissue of interest and lyse that tissue, and you know that in this region of the brain, cells express this protein at the cell surface. I don't know if you can do that in flow cytometry. You can kind of choose a region of interest within the brain and lyse that region to test for cell surface expression.
Okay. We have a question from Amelia in the chat. She says, thank you so much for your great presentation. I'm just wondering, for the validation techniques, did you have, like you had to do all of them? Is it common to do all of those validation techniques?
Yeah, like, so basically in my case, it depends on what questions you're asking. So I want to make sure that my protein of interest is targeted to the cell surface. It's the expression levels of that protein are not changing, and it's expressed in the region of interest with the mutation of interest. Because if that's not the case, we're trying to prove that it's just the homophilic cell adhesion that causes disrupting, just that causes the phenotype. So if we don't show all this, there's always a question of, well, is there a decrease in the protein levels? Is that why you're seeing the phenotype? Is the protein not trafficked to the cell surface? Is it similar to a knockout? Is that why you're seeing the phenotype? So is it not expressed in the axons? Is that why you're seeing the phenotype? So I think to prove the point that we're trying to prove, we had to do all these experiments. It depends on what you're trying to answer.
All right. Are there any other questions? If not, I have one final one. When you were talking about micro-injecting cells with Cas9, I mean, there's also like you can do electroporation or transfection. So I've never heard of micro-injecting to.
It's similar to that. It's very similar to that. It's like you can micro-inject the cytoplasm or the pronucleus of the cells. The literature shows that if you inject the pronucleus, the efficiency of this process is much higher. It's very simple. It's basically electroporation.
Okay. All right. So if there are no other questions, I want to thank you again, Neelima, for your presentation. We really appreciate you coming on.
Yeah, thank you for inviting me.
Yeah.